what we're going to look at is the flow of electricity around the cell. And we're going to remember the names, the parts of the names of the cell, right? Dendrites around here that are reaching out and getting signals from other neurons or whatever, or sensory receptors, as well as the cell body, right? So signals are going to flow in this direction, remember. It's going to be electrical signals coming from the dendrites toward the axon hillock, all the way down the axon into the terminal, the axon terminals to the next cell, right? Always keep this in context in your mind, because we're going to kind of get close in on a lot of these areas right here. All right, so when we're talking about the resting membrane potential, next we're going to talk about disturbances in that resting membrane potential. Right, so you have some kind of stimulus of some sort, maybe an incoming signal from another neuron, whatever. Right? This is going to change that resting membrane potential in some way. And it's going to be considered a graded potential. It's going to kind of passively spread around the dendrites and cell body, or it's going to be this action potential down the axon. And so we're going to kind of cover more detail about these types of two potentials. Basically, it's all change in the resting membrane potential, temporary change in the resting membrane potential due to stimuli. And this is the way cells communicate with each other. So when we're thinking about these two, graded versus action potential, your analogy for this, for graded potential, is if you throw a pebble in a pond right here, right, it's going to create a ripple right around the pond, big waves, relatively big waves around where the stone was thrown. And then as you move away from that point of contact right here, the ripples are going to get smaller and smaller as they move away from that source. Okay, that'll be a graded potential. For an action potential, the analogy is going to be a string of dominoes falling on each other, right? When this fall once, it drops and hits this one. The speed, right, stays the same from start to finish, right? There's no getting smaller of the potential right here. This one is just as big as this one down here, or this one rather, just as big as this one. So we'll get into the details of this, but that's your analogy we want to think of when we're talking about graded versus action potentials. So your graded potentials are a little ripple in the cell body and dendrites. They can be big or they can be small. They're variable strength. They're going to travel short distance and lose strength as they travel over the cell body and dendrites. There's going to be a stimulus with a big ripple. And then as you move away from that, that ripple is going to get smaller, right? This is all going to be due to an influx of ions, in this case, that are going to spread out, right? But as they spread out, they'll kind of leak out over time, and the signal, this wave, will kind of get smaller and smaller, right? So that's your graded potential. Reaction potential, again, very brief, large depolarizations, right? From resting membrane potential, you're going to get more positive, and this is always going to be depolarizations. Whereas this actually might be a so-called hyperpolarization. For your depolarizations, that is becoming more positive, this is going to very briefly, very largely travel for long distance, usually almost through the axon, let's just say through the axon, and it's not going to lose any strength along the way. Again, we'll get into more details, but that's just the overview of graded versus action potential. So to talk about these, we want to talk about these channels and this whole thing of moving down an electrochemical gradient. Right, again, they're kept out, they're kind of kept sodium and potassium in here, sodium out here, such that if they were allowed to move, they would move down their electrochemical gradient. Right? So for potassium, this is the example over here. If there was a greater amount of potassium ions over here, then over here, they're going to diffuse down their concentration gradient out of the cell because potassium was higher inside the cell. they would keep on moving out down their concentration gradient. But on the other hand, they have this opposing force, because remember, the inside of the cell was negative. At a certain point, they're going to stop diffusing out and kind of keep back in down their electrochemical gradient over here, their electric gradient over here, right? So they were positively charged ions. They're attracted to the negative, and then they kind of stop. And they stop it around when the cell gets around -70. And that's why the cell potential, the resting membrane, is -70, because they'll stop flowing out and the electrical attraction will be greater than their chemical attraction. So that's your electrochemical gradient. Mostly we're kind of be concerned about our electric ones because we're talking about sodium for the most part, but this is also a big factor. So when they move down their electrochemical gradient, they're creating a current, just like those electrons in copper atoms, right, going down a wire, right? They're creating a current. And for biology, again, we're usually going to be talking about a positive to negative as far as these electric currents are going, right? And as they're flowing, that's going to change the membrane potential. As these potassium ions leave the cell, they're taking that positive charge with it. So that's going to make the inside more negative as they leave. So the potassium ions are leaving and the cell's getting more negative. The cell doesn't want that, so they can close a gate. So all these ion channels are going to open and close due to the presence of certain types of gates that are going to open and close on a couple of different ways. And so it's a part of the channel that can open and ions can flow out, or it can close and ions are not going to flow out and keep them in. Okay, again, let's look at remind ourselves of the parts of the neuron, right? The dendrites, cell body, right, is going to receive the input signal from another cell. Along the way, in a way we'll talk about more next lecture, there's going to be an integration of all that signal, right? And if everything is right, it'll reach the axon hillock, and there'll be an output signal over here down the axon right here. Right, so for this part, we're going to be really talking about this first input signal on the dendrites and cell body right here for this graded potential. And then this part, we'll be talking about this action potential. So when I ask you about these ion channels, I'm talking about their location, where they might be found on a particular neuron. So we got these 4 types of ion channels that are important for your resting membrane potential, the graded and action potentials. So your leak channels are those ones like your sodium and potassium leak channels, mostly your potassium ones, which are much more heavily represented in the cell membrane of neurons. And so they'll be all over the place, right? And they kind of open, and this, your book is describing it as randomly opening and closing, right? Just based on whatever, but really there's a little bit of voltageness to them. Don't worry about it. They're just leak channels and they open and close, and potassium are going to flow in or out based on their concentration electrochemical gradient. And they'll be all over the cell. Dendrites, cell bodies, axons, all types of neurons. Next type of channel is going to open and close due to the binding of a molecule, right? Of a ligand. We talked about this for the neuromuscular junction, acetylcholine bound to a acetylcholine receptor, right? And that opened up the channel. It opened it up to, we talked about sodium, but in reality, any positively charged molecule on the outside was going to flow inside the cell down their electrochemical gradient. And then any things like potassium would flow out the cell when this is open right here in this example. Okay, these are ligand gated channels. Where they're going to be, they're basically going to be on your postsynaptic membranes, right under, just like we saw with the neuromuscular junction. And so these are going to be on the dendrites and the cell bodies where there's synapses. And for your central nervous system cells, your interneurons and your motor neuron cell bodies, they're going to be over there. But we also want to note that, remember, sensory neurons are getting their information from the environment. So their ligand gated channels, where they're stimulated, is way out in your skin somewhere. So your dendrites of sensory neurons are going to be out in the periphery somewhere. Okay, well, we can go back to that when we go over the types of neurons. Right, but these are ligand gated channels. Next one, we'll get to when we talk about sensory receptors and stuff. There's a lot of mechanically gated channels opened by mechanical stimuli. If your cell is stretched in any way, right, that basically just mechanically opens up the channel. We'll talk a lot about this when we get to conduction or transduction, rather, of sound to nerve signals, right? Because the sound waves are going to create these vibrations, open up this receptor, and create this electric flow. Okay, so those are them. And then the big one here is going to be voltage gated channels. These are going to be opened up by a change in voltage. So in this example, this is a sodium one. There's a certain type of voltage -70 resting membrane potential. If for some reason there's a positive charge running through here, this gate opens and then potassium starts flowing outwards. So these voltage gated ones, we're going to find them mostly near your axon hillock and your axons for the most part. This is where we're really going to kind of see these voltage gated, and they're really what makes a cell excitable, right? Because really any cell can have something like these around here, but your neurons and your muscle cells are going to have a bunch of these voltage gated ones. And because they are going to be what's opening in response to a change, right? This is going to be some kind of positive feedback cycle, or negative if it was in response to a negative charge, right? But these are voltage gated channels. Everybody clear on that, right? And we'll go over these as we move along here. Mostly we're going to really talk about these ligand gated and the voltage gated channels first. And so your ligand gated, right? These are like the acetylcholine ones. We already talked about the neuromuscular junction. They're going to be right under this postsynaptic membrane for the most part, pretty localized to this spot. Whereas your voltage gated ones are going to be traveling down the axon, right? Passing a signal on. And leak channels are basically going to be kind of everywhere, restoring your resting membrane potential in the event of a disturbance. All right, so that's it for the couple different types of ion channels and the distribution of them around a neuron. And next we'll talk about the currents that are generated by these ion channels and what they mean for cell communication. All right, see you next time.